Buenos días. Como primera ponencia del día de hoy nos acompaña el doctor Yohann Corvis. Él posee un doctorado en Ingeniería Físico-Química y en Ciencias de Materiales y Procesos. Su trabajo de doctorado se basó en la elaboración de superficies biocatalíticas mediante el autoensamblaje de sistemas bioorgánicos sobre superficies recubiertas de proteínas. Después de 2 años de estudios postdoctorales en la Universidad de Varsovia, la Universidad de Metz y el Instituto IFP Melmesón, fue contratado por la Facultad de Farmacia como profesor asociado en Físico-Química en el área de investigación de medicamentos. Allí obtuvo su acreditación para supervisar investigaciones en el 2013. Actualmente trabaja en el Grupo de Técnicas Químicas y Biológicas para la Salud del CNRS, INSERM, centrado en las materias primas farmacéuticas, caracterización de interacciones intermoleculares y formulación de productos para terapias anestésicas, antitumorales y antiinflamatorias, desde la preformulación hasta la evaluación in vivo de los sistemas formulados. Hasta la fecha es autor de 48 publicaciones, 3 patentes, un capítulo de libro y 2 reseñas de libro. Ha supervisado 49 pasantías y 3 estudiantes de doctorado. Su trabajo ha sido presentado en 66 simposios internacionales, 35 simposios nacionales y 22 charlas invitadas. Es miembro del Comité de la Farmacopea Francesa de la ANSM, de la Junta de la Sociedad Francesa de Nanomedicina desde el 2021 y miembro de la Junta de la Sociedad de Profesores e Investigadores de Fisicoquímica en Farmacia desde el 2013. Good morning. Today we're having Dr. Yohann Corvis, who holds an engineering doctorate in physical chemistry, material sciences, and process. His doctoral work dealt with the elaboration of biocatalytic surfaces by self-assembly of bioorganic systems on protein-coated surface. After 2 years of postdoctoral studies at Warsaw University, Metz University, IFP and Melmesson Institute, he was recruited to the School of Pharmacy of Paris as an associate professor in the physical chemistry of drugs research area, where he obtained his accreditation to supervise research in 2013. He is currently working in CNRS Inserm Group UDCBS, which means Chemical and Biological Techniques for Health, on state of matter of raw pharmaceutical materials, characterization of intermolecular interactions, and the formulation of final products for anesthetic, anti-tumor, and anti-inflammatory therapies. Reformulation to in vivo evaluation of the formulated systems. Today, he has authored 48 publications, 3 patents, 1 book chapter, and 2 book reviews. He has supervised 49 internships and 3 PhD students. His work has been presented at 66 international symposia, 35 national symposia, and 22 invited talks. He is member of the French Pharmacopoeia Committee of ANSM since 2021, board member of the French Society of Nanomedicine since 2021, and board member of the Society of the Teachers and Research of Physical Chemistry in Pharmacy since 2013. We welcome Yohann Corvis.
Thank you. Thank you very much for the invitation. I would like to thank the organizer of the Federation of Costa Rica of Pharmaceutical Students of Pharmacy students. So thank you very much for your welcome and the presentation. So I will talk today about the nanocrystals for cancer therapies. Why nanocrystals? And we will discuss about general nanomedicine properties and the future of those kind of techniques. Why the bioavailability of drugs are very interesting to deal with is because drugs are marketing as solid form because 80% of the API are solid at room temperature. That means that we need to improve their properties since the solid state has to be dissolved. For example, in the BCS classification system, so the biopharmaceutical classification system, more than 90% of drugs present low solubility, and at least 70% in the class 2 and 20 in the class 4. For that reason, we need to improve formulation of medicines to have better bioavailability, and we will see also better absorption in the tissue, accumulation in the tissue. It has been proved that around 0.7% of drugs can reach tumor environment tissues by a review submitted in Nature Review in 2016, and they showed by analysis 232 articles that around only 0.7% of the drugs were in fact accumulated in the tissues. So for that, we try to understand the reason. First, if it was a question of the nature of the drug, the organic or inorganic, the size, the charge, and also the active and passive targeting. I will discuss about this later. And it seems that we cannot improve, let's say, so much regarding all those aspects. And for that reason, we need to improve the carrier, the encapsulation efficiency, the stability over time, the vivo and vitro stability, and improve the release to achieve accumulation in tumor or complex environment. On the top of that, the encapsulation can improve systemic, reduce systemic administration of the drug by targeting the drug directly in the desired tissues. So to overcome those kind of problems, that means that bioavailability and accumulations, we need to improve physical chemical property of the drugs. For example, we can modulate the chemical properties of the active ingredient, like salt formation, creates solvates, or have a guest host assembly formation for bioavailability in this case, and also for accumulation. But in those cases, we have drawbacks like interaction which can be destabilizing and induce the accumulation of exogenous inactive compound in the body. We can play also on the physical modulation, so to reduce the drawbacks we saw with the chemical modulation by using physical modulation like polymorphism, amorphization. And in this case, you have cytotoxic effects due to the fact that it presents beta stability and thus can create, let's say, non-desired properties. And the final possibility is to reduce the size. The reduction of the size, especially the nanonization, can be interesting since it can have some properties that can be interesting for drug delivery. I will discuss this in few seconds. Generally, in the cancer therapy, you have different kinds of nanoparticles compared to the free drug. The free drug, for example, has a systemic administration, so can be very have side effects, very dramatic side effects in healthy tissues. And in this, for these reasons, we need to go further with targeting, passive targeting with the EPR effect. I will present this later then. Also, the active targeting, either the cancer cells targeting or the endothelial cells targeting to allow the drug to reach the cells, the cancer tissues. And I will discuss at the end of my talk about the release controlled by external and or internal properties. So you see that the science and pharmaceutical science mostly went to different properties from the free drug delivery until complex systems. To achieve let's say, candidates which can be useful for clinical use, we need to first find the originality of the drug, develop the production via all the physicochemical aspect and the optimization of the cost and so on, and also to present interesting in vitro and in vivo in animal models, for example, results. And I will discuss about the three first points, so the preclinical study. In general, nanomedicine has started since, let's say, more than 30 years. And I present you here a framework of everything was been done from 1985 to now with nanomedicine. So first with liposome in 1995 for doxil treatment of cancer. We see that we improve the, let's say, the technology of the nanomedicine until the nanocrystals around 2000, and I will discuss about this in a few minutes also. But also the COVID vaccine is belong to nanomedicine since it's solid lipid nanoparticles encapsulations of ARN. And you see that for this, you need to control, to adapt also the physicochemical properties, parameters, to efficiency via the structure, the size, and the coating of your nanomedicines. If we take a look about all the properties, so I propose you all kind of nanomedicine which can be useful for drug delivery. You can see that the size is important, for example, for biocompatibility, but also for clearance properties. So we have to deal with all those kind of parameters. The surface charge is very interesting in the COVID vaccine, also due to the fact that the ARN is negatively charged. So we need to stabilize it with positively charged lipids. The coating can be interesting, we see, due to the fact of the utilization to avoid the clearance by the reticuloendothelial systems, and the morphology also can be adapted to the wanted properties. In terms of circulation, so as I told, the reticuloendothelial, the MPS systems can't be, let's say, inhibit to avoid the uptake of the nanomedicine by the macrophages. And like that, you have an increase of the circulation times, which can be interesting, and accumulation of the drugs in the tissues, especially for tumor tissues, the vascular tumor tissues. So by preparing a nanomedicine which is adapted to avoid uptake by the macrophages, we can improve the accumulation of the drug in the vascular tissues compared to normal tissues. And this can be interesting for targeting delivery. So as I talk, we will discuss mostly about nanocrystals. Why nanocrystals? Because they present most advantages we cannot be found in other compounds, like tolerance, due to the fact that there is a high content of active ingredients, so with less organic solvents, so better bioavailability and let's say biological properties. It can increase EPR, DPR effect and reduce so the systemic delivery and the efficiency can be improved, and we will see the preparation is, let's say, with less steps compared to some other techniques. As far as the nanocrystal properties are concerned, they can be prepared via two different techniques. The objective is to have a specific area which is increased to allow the functionalization to avoid the recognition by the macrophages, as I told you about. You can also improve solubility and have stability which is comparable due to the fact that you are upper of the solubility, but you have to stabilize it with some stabilizing agent. For this, you have two main techniques, which are the top-down approach, like the milling, wet or dry milling, but also the high-pressure homogenizer. For those techniques, they are time-consuming. They can degrade your active ingredients or even your excipients. So for that reason, those kind of techniques were not considered for further experiments. And you have also the bottom-up approach, which consists from the molecular level to grow crystals until the nanosize properties. And the overall nanocrystals can, whatever the techniques that you have chosen, can be useful because they can improve the drug loading, so less administration to the patient. It can improve bioavailability, permeability, and accumulation. In vivo lifetime also due to the fact that it can be functionalized and have present sustained release properties and administration, various administration routes. The nanocrystal is the only active ingredient formulation that can be administered via parenteral or enteral route. And the overall is to improve the ratio benefit over risk. If we take a look especially on the timelines of nanocrystals, so the first nanocrystals were reported in 1978, so more than 40 years ago, and you can see that to have the first let's say the first nanocrystals cells on the market in 1982. It has been with gridiofulvin, which is an antifungal compound, and you can see that it was in tablets. So it was difficult at this time to stabilize into solution the nanocrystals, and it was for the oral route, but not, for example, for the parenteral route. And to have the first administration, you have in 1998 ocular administration, but to have the first one in a parenteral route, we have to wait 2009 with Invega Sustena, with the intramuscular administration in 2009. And then the Ragadenex in 2014. And for this, it has been, especially for the Ragadenex, extemporaneous preparations. So it has been solubilized just before administration. In 2021, and even in 2000, so less than 1 year ago, Cabenuva has been authorized by the FDA for antiretroviral therapies. And you can see that from the first nanocrystal to the last one, no cancer molecules, anti-cancer molecules for cancer treatment has been sold as nanocrystals. You should know that you have two nanocrystals which are right now in clinical trials, paclitaxel in phase number 3 and simetaxine in phase 2. And this was very interesting for us to try to produce nanocrystal for anti-cancer therapies. If we take a look on the annual deaths of cancers, we notice that around 40% of these cancers could be treated by etoposide, which is a topoisomerase 2 enzyme. Etoposide was for us our, let's say, a proof of concept for nanocrystallization. Why? Because we discussed with some doctors, and they told us that etoposide has some problem of stability issues. So at the hospital, and if we take a look on the different formulation of etoposide that you can find on the market since 1993, you can see first that you have a solid dosage form with high amount of etoposide, so for systemic delivery. You have the liquid form for the IV route, but it is a solubilized form thanks to 33% of ethanol prior dilution. And you have also in 1996 the solid dosage form, which is used for IV route after extemporaneous here or also preparation. So all those kind of different formulations showed to us that etoposide in the hospital has some drawbacks in the stability issues. First, of the solid form, is of the liquid form, because it's the reason why they want to go through the extemporaneous preparation. The variability in the pharmacokinetics due notably to the solid dosage form, and side effects due to the fact that most of them have, let's say, systemic delivery. Here I present you the 7th pharmaceutical approved compound of etoposide, and we will work mostly with toposar for the next slides. So for all those reasons, etoposide showed to be a good candidate for nanocrystallization. As far as the in vivo properties of etoposide is concerned, you should know that etoposide eliminates topoisomerase 2. That means that this topoisomerase increases the split DNA complex, and by stopping this action, it leads to apoptosis of cancer cells, since the topoisomerase is overexpressed in cancer cells. But the fact that topoisomerase is overexpressed in cancer cells make the topoisomerase also produced in healthy cells. Is the reason why nanocrystallization could be interesting, since non-specific mode of action of etoposide is concerning especially cancer tissues. For these reasons, so we try to work with etoposide, and the log of P coefficient of 0.6 was interesting for us, because we have patented a property which is essential for the nanocrystallization, is that the log of P should be between 0 and 3. Another nice property of etoposide is that it can be used, and it has been proposed by Eyagi in at the end of 2019 with the apparition of the coronavirus COVID-19. It has been proved that etoposide with an anti-inflammatory compound can improve lung injuries by reducing the lung injury score when it is associated etoposide and anti-inflammatory compounds. And this is very interesting, and it has been then proposed by Tamaki in 2020 that we can use etoposide to cure COVID patients in the high level of disease, due to the fact that it can play on 3 different parameters: the inflammatory cytokine production, which can be reduced, suppressed, so reduced. You can also activate macrophages and eliminate the macrophages which has been activated, and you can activate the T cells, cytotoxic T cells. And for these 3 different parameters, we can propose to cure patients which are severely infected by COVID-19. As far as the optimized protocol is concerned, so the nanocrystals were prepared via the anti-solvent precipitation. That means that you dissolved your drug in an organic solvent and then disperse it in water. The originality of our techniques is to, for the first step, we were the first to not use stabilizing agent in this step. So it's the reason why we obtained patent about this kind of preparation. Then we evaporate the total liquid, so under vacuum with a standard procedure that we developed also in our lab, to obtain a dry powder, which can be redispersed in stabilizing agent aqueous solution. And the stabilizer agent is inserted at the end of the preparation with sonication. We first tried to optimize the ratio of etoposide over the stabilizing agent. Here we choose the poloxamer P407, which is also called Chloronic F127. This is a polymer which is FDA approved, and it is famous for the stabilizing agent of liposomes, for example, or other crystals. And we found that the optimal ratio is 1/2 etoposide P407 with size which is comprised between 1 and 200 of nanometers. It has been confirmed also with the DLS, so with light scattering experiments, and we even shown that the optimal organic solvent over water for the first step of the anti-solvent precipitation is 1.5/10. And here we showed that if here you have the ratio of the stabilizer, so before the evaporation, so at the step 1, you see that the ratio before and after, so is the best if you have no polymer before, you have a good size, let's say less than 200 nanometers, and it is stable over time. And on the top of it, you improve the yield of production since you don't have polymer in the first step, so you don't lose compound due to the fact that you have bubbles during the evaporation of your solvent. And this has been patented in 2018, and we obtained in 2020, so 2 years ago. Different parameters were followed to understand first the nanocrystallization and to confirm it also. First, in the dry powder, so prior to rehydratation of your drug, we use the electron microscopy techniques, also the thermal analysis technique, and the list of the compounds. For example, for the thermal analysis, we reduce the melting temperature, which is the case for reduced size of organic compounds. In the aqueous solution, we made electronic also electron microscopy, so for TEM experiments, and X-ray diffraction in collaboration with the BC Cancer Agency of British Columbia University in Canada. And we showed that we conserved the nanocrystals in the solution even after the stabilizer agent. It was not so obvious because you have two kinds of problems here. Is that with the stabilizer agent you can form, for example, a kind of emulsion, which is not a nanocrystal by definition, or you can also create amorphous compounds. But here we showed that we conserve the nanocrystal properties for our step of production. Then we went further with dialysis to see if there was a sustained release of your drug over time. And for this, we used as a comparison Topozar, so the commercial etoposide compounds by Teva, the nanocrystal, which is in red, and the microcrystals that's in the raw material in black. You can see that we, compared to Topozar, we have dissolution speed which is lower with the etoposide. That means that with the nanocrystal we can reduce the solubility, you can sustain the solubility of your drug over time, and compared to the microcrystals, you improve it since you reduce the size, you have a higher solubility, rate of solubility. I just want to tell you here that we don't have 100% of release because in this site, because we are not in the same condition. So from this time we decided to work in the same conditions, and we repeat the experiments, and we reach the 100% of release, and we have the same difference between the Topozar, etoposide, nanocrystals, and microcrystals. So the dialysis proves first that you improve the speed of dilution, and you have a sustained release, a release over time. Then we went for cytotoxicity effects, so in vitro experiments on two different cells, the CT26 and the 3LL cells. So the first one is the murine colon carcinoma cells, and the second one, the murine Lewis-Lung carcinoma cells. And we shown that compared to the control, you have apoptosis of your cells when it's incubated with etoposide, and you can even see some nanocrystals of etoposides in the cell, mostly in the cytoplasm. The IC15 were also determined. We showed that there was not a big difference in the I say 16 if you compared the 3L, if we compare the nanocrystals and the free etoposide, but there was a difference with the toposar. Then we work with also hospital Gustave Roussy in Paris, and we showed with them that in the mice, if so we inject the equivalent of 10 milligram per kilogram in the mice, that you have improve of the pharmacokinetics at least during the 10 first minutes, due to the fact that you have a high amount of drug which is not eliminated compared to the toposar, because this was indeed with the same amount of drug injected to the mice. And this can be very significantly proved with the AUC, so the area under the curve measured during, let's say, during the 2 first hours, and we showed that you have much more toposide, let's say, circulating in the mice compared when it is in the nanocrystal form compared to the toposar in blue form. On the top of that, we try with a student of pharmaceutical studies in Paris, so the 4th year, the 4th and the 5 years, to calculate the parameters of the pharmacokinetics, and they determine half time of around 6 minutes for the alpha half times, and for the elimination of around 45 minutes, and for the etoposide, and improved compared to the toposar. This will be published soon in the local journal of the pharmaceutical student of pharmacy in Paris. The bio distribution also of etoposide has been determined. We proved that the nanocrystals were most, let's say, accumulated in liver compared to the free etoposide, and compared to toposar, and in the lung, it was the opposite. The toposar was highly accumulated in the lungs compared to etoposide nanocrystals. And in the kidney and the bladder, there was no accumulation at all of etoposide. Then we went for the vivo, let's say, in vivo properties and mice survival over time by following the mice weight and tumor size, and also the survival rate after 4 injections of 10 milligrams per kilograms. As far as the tumor volumes is concerned, we showed that with nanocrystals in red, we can significantly reduce the tumor size progressions compared to the toposar and the control. What is interesting is for the toposar, we had mostly the control evolutions, due to the fact that in the toposar you have an amount of ethanol which can be not be so say good for the tumor evolution. As far as the rate is concerned, the rate of the mice, so we saw not quite difference, except for toposar, where after the 4th injection, you have loss of weight compared to the control and the nanocrystals of etoposide. And for the mice survival, we showed that we can delay let's say the IC15 of more than 1 week for those kind of studies. If we compare the control and the toposide to the nanocrystal of etoposide. So all those experiments first confirm us the utility of nanocrystallization to cure at least cancer. For the thought of the project of nanocrystallization, it seems that so we have some small size compared to, for example, the top-down approach that I discussed before. We try to obtain very good polydispersity. We can go until 0.2 of polydispersity, and at minima we can reach 0.05 of polydispersity, which is very interesting for nanocrystal and for pharmaceutical application of the nanomedicines. The fact that we don't use a polymer is very interesting for improve the yield, as I told you about, and we show also that we can improve the stability over time directly in the solution. That means that no need to perform extemporaneous administration. And what is very interesting is that we can generalize, and this was our point of the patent, the feasibility of this technique for different kind of drugs, but also solvents for the anti-solvent precipitation. So for ongoing experiments, so we are working right now on those kind of experiments. We try to use different kind of therapeutic agent, like anti-inflammatory, antioxidant, neuroprotective agents. So here I showed you some kind of nanocrystal solution that we obtained for each of them. So from etoposide, ficetine, for example, ibuprofen, curcumin. And we want also to understand and control the crystal growth with different parameters. So the improvement of polydispersity, I told you about this, we achieve this. The different stabilizer agent, this also is under investigation, but we have nice results. And different kind of parameters that I will develop in few seconds. And we want also to upgrade the technology to active targeting, which is the case of a PhD student who is working on this in collaboration with Collège de France for the fellowship, and the scale-up production in collaboration with the Taiwanese and the Chinese lab also, and South Africa lab too. And we want also to work on immuno and chemotherapies, mostly by collaborating with the deep tech industry. And we will develop also diagnostic application, and I will also put you some results at the end of my talk. So for understanding the control of the crystal growth, thanks to Panma, which is in PhD, we optimize each step of the preparation of the nanocrystals. So we try to optimize each step of them in terms of protocol, content, concentration, and time, necessarily the decompression rate time. And for this, we drastically obtain very nice and reproducible results. We work also to understand our nanocrystallization to have a project with École Normale Supérieure of Paris, INS, work with microfluidics, to reproduce with microfluidics the preparation, so with the anti-solvent and the solvent, we can produce here the nanocrystal and detect it by different kind of device like light scattering, but also fluorescence microscopy. And this is very, and X-rays also. And this is very interesting since we can reproduce in situ the nanocrystals and analyze individually each nanocrystals. And in collaboration with Central Supélec, we use another technique which is famous in our lab too, is the non-photochemical laser induced nucleation, LPLIN device. We can, thanks to laser, induce the nucleation without touching the chemical properties of your drug. So this, we can also produce nanocrystal by this way and compare it to our protocol and see what is the advantage of each kind of protocol. We work also to understand well the organization of the drug on a new technique, which is the cryo-microelectron diffraction. And like that, we can target individual nanocrystals and learn is structure, exact structure in the solution. And this is very also interesting to understand the world of crystallization. So here I show you new experiments on fisetin. So fisetin, which is interesting and a model, let's say, drug, since it can be used for several pathologies, like anti-cancer properties, but also antioxidant and neuroprotective properties. So you can see here the shape of the nanocrystal that we obtained, so around 130 nanometers, and we obtain very interesting polydispersity, and we showed that with the determination of the zeta potential that the stability were due to steric properties of the peroximer, so the stabilizer, and non-electrostatic properties. This was a very interesting conclusion, and I didn't write it in this slide, but the pH also was 7.4, and for this kind of preparation, the pH is around between 7.2 and 7.6. So it's very useful to use zero properties of pH. We also try to understand, as I told you before, the storage condition. So at 5 degrees, so in the fridge, but also after freeze drying. So we use for this a cryoprotectant, which was mannitol, and we showed that we could, with mannitol, let's say, keep the nanocrystal property, even if it slightly increased in size, and the PDI was the same mostly. For the cumulative release, here we are in the same conditions. So for this, we still make some experiments. We are making some experiments to understand why we don't reach after 2 weeks the 100% of release. So maybe we have a very, very low and good sustained release. So we have to make more experiments about this, and they are ongoing. And we prove that with confocal images that we have nice fluorescence properties of the fisetin nanocrystals. And with fluorimetry, fluorescence spectra analysis, we showed that we have induced aggregation enhanced by the fact that you were nanocrystallize the ficetine. So the nanocrystal for fluorescence were very important for fluorescence as nanocrystals compared to bulk. The cytotoxicity also is ongoing right now, and we showed with those two cell lines that I present you, no, not the two, the 3LL that I present you, but the human also EAHY926 endothelial cells. We showed that we had a slight, let's say, influence of the ficetine compared to the free ficetine when it is in the nanocrystal properties. So to go further, we want to, as I told you, work on the active targeting of the nanocrystals. So we are working right now in collaboration with another lab of University of Paris on the modification, the functionalization of the nanocrystals by using chemistry of antibodies to target specific cancer tissues. And we want also to develop new smart properties of the nanocrystals by make nanocrystals that only activate in the acidic environment of the cancer environment, mostly due to the fact that we use hyaluronic acid, which can be cleaved and link the drug in pH around 5 compared to normal pH. And this is in collaboration with the Belgian company Materia Nova, which financed also the PhD on it in the lab. And the last smart material that we are working on is to use this time not a local intern stimulus, but an external. When you inject the nanocrystals in a liposome which is thermosensitive, we can, via the application of local heat by ultrasound, for example, to link the drug, which can be active in a cancer environment, and the ultrasound can release your drug, but also, let's say, perturb the endothelial cells and the permeability improve of the cancer cells. So what I show you today is that by functionalization and engineering of nanomaterials like nanocrystals, you can improve the loading of your drug, use several various administration routes to treat patients via the control bioavailability and stability, which is very interesting in vitro. And this is a part of a special issue that is now in submission opening. So if you have some paper to submit concerning supramolecular nanomaterial for engineering, you can submit it in the frontier of chemistry with this guest editor, which are present here. And there is also another paper, another article, which is open for submission concerning this part of formulation of solid materials to improve their bioavailability in pharmaceutics until April 2022. So I would like to thank all the people who worked in the lab and in collaboration with us for the nanocrystal properties. First, Bryce Martin, which is now in postdoc in New York, in Weill Cornell Hospital, and he developed all the first aspect of etoposide. I would like also to thank Pan Panma, which is a China Scholarship Council PhD student in the lab, and she developed all the generalization, the optimization of the nanocrystal formulation. Ferras, which is a PhD student also in the lab right now, and is in the first year, and is working with the Collège de France to develop the pH-sensitive nanomedicines. And the different students which are internship in the lab. Luis, which is from Costa Rica and is now in the lab, is in student in Nanomed, Master 1. Giovanni and the other one, and Jean-Claude from Malta, who worked also on this project. The BC Cancer Agency, I told you about it. Gustav Russi also, INS also, Matea Nova and Central Supélec. And if I have time, I would like to say to you that you are welcome at our lab if you want to work on nanomedicines. And I will present shortly some axes of work in our lab. The first one is the one that I present you today, the encapsulation of nanocrystals, for example, for thermosensitive application. But we have also the hydrogel formulation for a local application of your drug and delivery of your drug directly, locally, after chirurgical application. We work also on microfluidics to propose optimization of protocol of formulation of nanocrystal and also liposomes. And since 2 years, we are working also on design of experiments and artificial neural network to optimize our formulations. As far as nanomedicine stabilities are concerned, we worked also on RNA messenger vaccine, so since a few weeks, to understand the stability under notably cryogenic properties. And we work also on nanobubbles for gene delivery, but also for echography improvement of resolution. As we work also on nanoparticles and barrier crossing by the oral route for siRNA tablets. On the passage on the placenta of drugs, if we can't use, for example, liposome, if we want to treat the baby, we use some kind of liposome. And if we don't want to treat to the drug to reach the placenta, but cure the future mother, we change our liposome. So this is the work of Karine Andrieux in our lab, and we work also on the anti-neurodegenerative therapies, mostly for ischemia cure. And the last part is the bioluminescence property for theranostic applications. So this is mainly by Yohann and Cyril Richard, and we work with this with fresh NMR IRM medical images by magnetic resonance to localize directly in the mice the drug. So this is very interesting for us. And the reason why that we work with C18, for example, is to go on with theranostic application. That means that the association between delivery and diagnostic to see if the drug is reaching the cancer tissues, for example, for C18. And our lab is in Paris, so I would like to present briefly our lab. So Paris is the capital of France, or the city of the lights. We are located here So here is the pharmaceutical building, and we are close to a nice place in Paris, the Jardin du Luxembourg, Notre-Dame de Paris, which is here, Bastille, which is famous for the Revolution, Tour Montparnasse, Montparnasse Tower, which is famous because it should be a twin tower like in New York, but finally they made one. You have your Concorde Place, Louvre, Eiffel Tower, of course, the Invalides, and le Grand Palais, which is famous for exposition. So we see that we are close to a very nice place in Paris. And as far as concern our faculty, we have around 4,000 students from the first to the sixth year of pharmacy, 200 professors, 6 facilitators, 40 laboratories, one botanical garden, which is here, which is very nice to USABS. It's just here on the front of the botanical garden. And we have also one big central animal house. This School of Pharmacy was created by Napoleon in 1804 and has moved in this current place in 1882. And it was one of the first pharmaceutical schools in France with Strasbourg and Montpellier, so in 1804. In our building, we have a very famous salle des actes where the students present their PharmD diploma oral. And this it's an old fireplace. It is very interesting because it's classified in the beautifulest monument of France. And you have all the pharmacists which were famous, like Antoine Auguste Parmentier, which is famous because he bring the potatoes from China, from Asia to Europe under Louis the 16th. And he was famous because he was a pharmacist and an agronom too. And the other famous one is here, is Henri Moissan. Henri Moissan, which is famous because it was in our university, but it was the first French prize, Nobel Prize of chemistry, and till now is the only pharmacist who won the Nobel Prize of chemistry. And he has the Nobel Prize in 1906 because he made the big discovery, the isolation of fluorine. And he worked also on diamond. He prepared diamond in the lab from graphite. And our lab is in part belongs to his whole lab, so we are a little bit proud of him concerning this. So I would like to thank you also for your attention, and the different students that I told you about are there, Pan and Ferras in PhD, Jean-Claude from pharmacy in Malta, and Nathalie, which is the head of our group and care, which is associate professor with me. And thank you for everyone for your attention. If you have some questions, I will be pleased to answer.
Thank you very much, Dr. Corvis. Now we're going to start with the Q&A session.
I have a question in the chat. Does the slow rate release of nanocrystal, as in the case of paclitaxel, not influence the cytotoxicity of the same? All right. The point is that, and this is a very interesting question, is that the slow release should, let's say, begin once the drug is in the tumor tissues. So this is the deal, and is the reason why we try to encapsulate or to use, let's say, internal or external stimulus to release the drug directly in the tissue environment. That means that in that case, it should not influence the cytotoxicity, but you can have a longer effect of the drug in the tumor environment. I hope that Amonio, I answered to your question.
Thank you very much, Doctor. Now we proceed to see if anyone of the presential audience has any questions.
Hello, it was very interesting. I have a question. Is it possible that new drugs with those nanocrystals are marketed in the next 3, 5 or 10 years? Est-ce que c'est possible que des nouveaux médicaments avec ces nanocristaux soient commercialisés dans les prochains 3, 5 ou 10 ans?
Thank you for the French question too. As I showed to you, you have already nanocrystals. For example, in 2021, you have the Cabenuva, which is an antiretroviral agent, and it is two formulations. So you have two administration at the same time to cure, and so there will be more and more nanocrystals. I showed to you also that you have paclitaxel and timectaxine in clinical trial. So we have to follow what will be the final application, if it will be approved definitely or not. And for my nanocrystals, so let's say that now we are approached by different companies who wants to develop the nanocrystals. So now I think if we succeed, we have to wait, I think, from now a little bit less than 8 or 9 years if everything is okay. A little bit less, let's say 6 to 8 years if everything is okay, or the green lights are on for our results to have our nanocrystal on the market. But for now, I don't know, since we need for this to go on with the clinical trials, and for this you have a regulation aspect, and we are working on that right now with the different company to follow. So it's we have to work on that, but we hope, let's say.
Merci beaucoup. Thank
De
you.
rien.
Thank you very much, Dr. Corvis. Now I give the pass to Jimena.
So we're not having any more virtual questions. Thank you very much for this great presentation. We appreciate it very much. And it's very important for the future to know about this. So thank you for letting us know about nanocrystals. Thank you very much.
Thank you. And